You can start, sir. Sudhir, sir.
Yes, sir. A very good morning and warm welcome to all for this 8th session of AICTE Atal-sponsored online FDP on Nano Furtherance of Pharmaceutical Engineering and Industrial Pursuit, organized by Department of Pharmaceutical Sciences, Nirmala College of Pharmacy. I, Sudhir Madhyala, consider it a great honor to introduce about the 8th session resource person, Dr. Suryakanta Swain. Dr. Suryakanta Swain is presently working as a founder principal and head associate dean, School of Health Sciences, comprising 14 different programs and 9 departments under the Assam Kaziranga University, Jorhat, Assam. Dr. Swain recently established 6 laboratories infrastructure, increased 6,200 intake capacity of B.Pharm course, and introduced lateral entry for B.Pharm course and 4 new emerging PG programs. SAR designed the course structures and syllabus for the need of the northeastern region of India based on course program outcomes. Dr. Swain recently applied for 2 research project grants as a principal investigator under the Science and Engineering Research Board and BIROC of worth rupees 50 lakhs. It is now considered under Northeast Scheme. He completed his PhD from Berhampur University, M.Pharm from Bizu Patnaik University of Technology, and B.Pharm from Berhampur University, Orissa, by qualifying NIPER. So far, he has 13 years of teaching, research and development path from a higher level academic administrative role as an associate dean, principal, vice principal at different universities and institutions. Dr. Swain published more than 100 research, review, editorial, opinion articles of Scopus, SciIndex, PubMed, JCR, UGC Care Listed, peer-reviewed international and national journals of Springer, Elsewhere, Taylor and Francis, Informa Healthcare, Bentham, Dow Press, etc., with the highest impact factor of 7.09, with more than 1,500 Google Scholar citations and 750 Scopus citations and a 26 ResearchGate score, with a core research domain area of quality by design enabled formulation design, optimization, and characterization of mucoadhesive self-emulsifying gastroretentive transdermal nanoparticles drug delivery systems. He has edited 6 national textbooks as per the new PCI syllabus and of M.Pharm and B.Pharm courses, and published 7 international textbooks and 35 national book chapters as an author, co-author of reputed publishers such as Springer, Elsewhere, etc. He received 1 Indian patent grant and published 1 Indian patent from the Ministry of Commerce, Government of India, and patent in innovation already initiated for technology transfer with the pharmaceutical company. So far, he is the permanent advisor, editorial, reviewer, board members in more than 100 national and international journals. Dr. Swain is member of Board of Studies, Academic Council, IQAC, UDRC, NAC as a chief school level NBA coordinator, R&D, external expert, member of selection committee of various universities such as the Assam Kaziranga University, Acharya Nagarjuna University, JNTU Kakinada, JNTU Hyderabad, Annamala University, Bizu Patnaik University of Technology, Berhampur University, Geetham University, Centurion University of Technology and Management, Singhania University, Amri University, etc. Currently, he is guiding 10 PhD research scholars, guided more than 60 M.Pharm, 40 B.Pharm and 7 PharmD students. And he received several awards such as Emerging Leader in Health and Medical Sciences 2018, Professor M.L. Corona Memorial Award, Rajnibai V. Patel Pharma Innova Competition Award, etc. Dr. Swain delivered and attended more than 100 AICTE, DST, DBT, PCI, ICMR sponsored at national and international seminars, conferences, workshops, FDPs, SDPs, STTPs, webinars as an invited speaker and resource person. He is the life member of several professional bodies such as FIC, APTI, IPA, HAB, IPGA, ISPOR, and members of reviewer MS Research Australia, Research Management Council Australia, member of Healthcare Advisory Board Canada. Before inviting the resource person, I would like to inform all the delegates to post the queries in the chat box. At the end of the session, queries will be clarified by resource person. Now, I would like to welcome Dr. Suryakant Swengaru to start the session on showcasing the quality by design framework on self-nano emulsifying drug delivery systems.
Yeah. Thank you very much. Thank you very much, Sudhi sir, for your nice introductions about my brief past academic teaching and research experience. Thank you very much to introduce me to all the participants across the India. Thank you very much.
It's my pleasure, sir. Thank you.
Before to start my presentations, it is my heartfelt gratitude and honors to say my thanks to the Nirmala College of Pharmacy Management and the evergreen dynamic principal of Andhra Pradesh, Dr. Sheikh Abdul Rahman sir, for inviting me such a beautiful e-platform to deliver my talk on quality by design showcasing for industrial perspectives, basically self-emulsifying drug delivery system. And I also congratulate the coordinator of this program, Dr. Reddy. He is coordinating the program in a nice way, and the team of entire fraternity team of Nirmala. Really, you are doing the commendable job in this pandemic situations and arranging across the India and different resource persons from different institutes to deliver their past experience to gain the upgrading the knowledge of the new faculties who are starting their careers in the teaching and learning process. So thank you very much for giving me the opportunity. And before to start my presentations, I will say the few things, then immediately I will share my slide to all the delegates as well as the participants. Dear friends, nowadays it is very, very difficult to sustain anywhere or any field, whether it is teaching or research or in industry, whatever the domain you are working. The difficulties is that how extent you know about your core field of specialization. If you are expert in the field of your domain, then definitely there is a chance of sustainability for long time. Otherwise, it's very, very difficult to sustain in any institution or any research organization for long period of time. So quality is the very, very important for our day-to-day life. Quality is always compromised only by design, not by chance. If you think that you will do something by today and tomorrow same thing you will not continue, then there is a chance of question mark. So to avoid such question marks, then you have to plan properly or you have to execute your plan according to your objective, and based on your objective you can find out the best possible way of your your job profile. Then only you can reach your destination without any problem. So this is my first advice, and later on I will show you one by one slide and I will go through my presentation without your wasting your valuable time. Shall I share my slide?
Yes, sir, you can share, sir. Already I have given the access.
Okay. Now my screen visible to you, sir?
Yes, sir, it's visible, sir. Yes, sir, it's visible, sir.
Okay. Yeah, dear friends, the topic chosen by Nirmala team is really a beautiful team. Earlier, most of the scientists or the research professors or academic professors, they are keeping the theme which is not relevant to the current scenarios. Industry nowadays, need the product which is really developing in the academic field. What actually we are doing? in our academic field that should be having, industrial applications. The advancement of nanotechnology in industrial point of view, if you are developing something in your academic research, then there is some kind of values. So AICT is definitely choosing a very, right project and given the, sponsorship to the Nirmala institutions under the scheme of AICT training and learning, because without developing our teaching and learning scheme, will not sustain. I already told you from the very beginning, we have to upgrade our teaching and learning process, including the research. So the theme is very, very pertinent for industrial point of views. What I have chosen, that is focusing of the quality by design framework on self nano emulsifying drug delivery system. Basically, why I have chosen, because it is having some industrial scope or industrial applications. And if you go for, QbD, the pharmaceutical quality by design concept, QbD, it is not a buzzword, Nowadays, it is most of the industries giving the first priority for pharmaceutical product development as for ICH or US FDA guideline. The concept of QbD is applicable everywhere and gained, significant, attentions globally, not in, nationally. It is having globally impact is there for the development of pharmaceutical product, basically, in commercial batch productions in industries. And the key behind the QbD is, the implementation of QbD is very, very important. How you want to apply the QbD software or the design of experiment software during the manufacturing process to maintain the standard of, drug product as regards to efficacy and, potency, to get the quality product or robust product, or you can achieve the high therapeutic efficacy of the product, then you have to apply quality by design concept. Dear friend, ICH Q8 guidelines, the concept of QbD is, applicable systematically, and it is risk-based product developments using predefined objective. What is your objective during the process development or during the manufacturing operation of your pharmaceutical drug delivery system in order to get a quality, robustness, and to achieve your desired product performance. And this QbD is always, it is not a simple way to apply. If you have the knowledge about the QbDs, then only you can convert the QbD into a simpler form or simpler product developments without, regulatory issues. Basically, when you go for product filing for approval. So in order to minimize such regulatory problem, then you have to apply quality by design concept in your drug product development during the optimizations process. And I told you, quality by design is not by chance. It is by proper planning, proper executions, and you have to find the, the parameters. You have to shade the QbD parameters and what are the quality by designs parameter really required for a different drug delivery system. The key parameter you have to set, what is CPP, what is a critical process parameter, what is critical formulations attributes, what is qualitative target product profile, what is QTT, so what is only target profiles. All this you have to set it before design your formulations optimizations. Friends or dear delegates, for you developing your formulations, let us plan properly, and you, according to your plan, go for certain trials. After manufacturing, developing certain trials, then only you can develop a very good formulations. So this is the part of introductions or background of my presentations. Today presentation, I'll go one by one. I told you, applications of quality education is very, very important, to get feedback from the primary stakeholder, from your institution or from your organization. Yeah, the primary stakeholder of any institution or organization, the students. The students always judging the quality of teaching, quality of your, what quality you have during your classwork time or class time. Every teacher, needs to improve, not because they are not good enough, but because they be even better, according to the statement of Dale and William. We are every day upgrading our knowledge by attending, different webinar, different seminars, symposiums, workshops, irrespective of our domain, whatever, just upgrading our knowledge. Once we upgrade our knowledge, then definitely this knowledge we have to implement in our day-to-day, job profile, that is teaching learning process. And basically the students will judge what quality you have. If you are not caring to the students, if you are not nurturing to your students, and if you are not connecting with your students with your proper educations or your proper knowledge sharing to the students, then you will not become a quality teachers. For every quality teachers, quality education is required. Similarly, in your pharmaceutics or in pharmaceutical drug delivery system, if you want to develop a quality product, then quality by design's concept or application of QbD or FBD or DoE or optimizations concept are essential in order to develop your skills or your formulations optimizations without having problems. So what about my presentations contents to be discussed for today's presentations? That is, I discuss about the optimizations process. What are optimizations? DoE concept, that is design of experiment, quality by designs, BD formulations by designs, and experiment by design. I'll discuss one by one. And what are the different types of design space and variants of design space, and what are the regulatory importance in pharmaceutical industries, and what are FDA 4-point industrial strategies, design space with confidence intervals and its desirability functions, and what are the computer-based software available now freely and paid versions in order to optimize your different trials in different drug delivery system. And I will discuss one or two case studies related to self-emulsifying drug delivery system based on QbD or FBD drug delivery system. Dear friends, the earlier director of CDER, that is Center for Drug Evaluation and Research, FDA, what she said, quality never compromised, and you have to give quality is the first priorities according to the J.M. Jaran concept in the year 1990. What the Jaran, he is the quality expert. He given you have to give the first priority of the to the quality person, not to the quality by testing persons. You have to give quality persons first priority according to trilogy of quality by designs. And in order to get a maximal, efficient, and agile, flexible pharmaceutical product in manufacturing sector or in pharmaceutical industry, globally, if you want to get your product with high quality performance without extensive regulatory oversight, then you have to change something about your product during the manufacturing operation by the applications of QbDs in order to get the desired product, or you can develop a pharmaceutical product without applying QbD. It is not at all possible. According to her statements, that is Janet Woodcock, she said you have to apply QbD concept or QbD software during the formulation optimizations process. What formulations we are developing or what kind of delivery system, it is not easy to apply immediately without knowing the raw materials, what are the process you are adapting, or what are the techniques you are using, what are the resources you are utilizing, and what are the infrastructure you have during the product development. Dear friends, any drug delivery system, it is a very, very complicated drug delivery systems, because we are using single API or sometimes, multiple or combinations of drugs for development of different kind of delivery system using, various bunch of or plethora of excipients or polymers, and the polymer or excipients having different characteristics. Sometimes we are developing rate controlling drug delivery system or control drug delivery system or sustained drug delivery system. We are selecting rate retarding polymer like HPMC or EC, and how the EC or HPMC impacting to the drug. Before that, we are doing some certain kind of characteristics pre-formulation studies. So like that, how you have to select and what kind of polymer is required for what kind of delivery system. In order to balance a suitable drug delivery system, then you have to apply the optimizations process, or you have to apply the optimizations techniques to get or to prepare your own drug delivery systems, which giving the desired characteristics according to your selective objective or your pre-planned objectives. So if I'll say a beautiful example for your day-to-day life, it is not that much easy to take the optimal decisions. Your friends, sometimes if I'll give you two things, one I'll give, slippery dish, and I'll give, delicious ice cream. But two options you have, which one you have to select and which is best for you. No, somebody will offer you good position in any institutions or organization with less salary, and somebody will offer good salary but low cadres. So which one you have to select? It's very, very difficult to take the optimal decisions. And in order to satisfy your boss, satisfy your authorities, because different persons have different needs now, different ambitions, different objectives. And can you compare with for this example, this slide, that is delicious ice cream versus scruffy silky dress, which is very fit for you, whether ice cream you'll take, and what are the advantages and what are the impact to your bodies, and the dress, what is impact on your looking. So everything is depend on you, how you have to choose your vitals characteristics or your elements, basically QbD elements, which is having influential characteristics during the product development. So it is not that much easy, confront different situations as diverse duties to perform, different persons to please with dissimilar needs, demands, taste as mad options to choose from. How you will select? The selections is based on the preliminary trials, preliminary observations, and you have to find what are the advantages, what are the disadvantages for these two things. If you will sort out the best possible parameters among the several elements which is having greater influential activities or greater influential performance, desired performance for the product developments, then only you will be happily balanced your drug delivery system and a content manners. So you have to choose the vitals among the several trials, several experiments with if you apply the optimizations techniques, then you can minimize the number of trials, resources, time, efforts, manpower, so many things. So optimizations you have to apply rationally in order to get your optimized product in a balanced way. So the optimizations comes into the existence. So it is nothing but key to keep things in a balanced, orderly manners. So if you want to balance both the arms, then you have to select what is your independent variables and how the independent variables affect to the response. So the selections of both the sides, dependent and independent variables, is very, very essential at the time of applications of optimizations techniques. In order to optimize your product rationally, or your entire pharmaceutical product batches, then you have to apply design of experiments, DoE software. It is a rational approach by which you can find out what is the characteristics about your drugs and what is the behavior of the selected excipients during applications of different techniques or manufacturing process or manufacturing different techniques during the development of your products. So that's why always you have to apply DoE concept in order to get the robust quality product, which are quality batch or quality formulation, in order to go for gambling of your formulations by heat and trials method. Don't go for 100, 200, 15, 20, 25 or 50 formulations. You have to waste the chemical solvents, manpower, time, etc. So to optimize all these things to get into a true optimum product or the batch, then you have to apply DoE software or to get your robust products. So the DoE concept, if you want to apply, then you have to define your DoE objectives and you measure the applications of DoE. Analyzations of DoE software is very, very important. If something is giving some bias or some not significant data you are getting after analysis, ANOVA of different parameter or different responses in the software, then you have to improvise to change the coded labels, or change the selections of independent parameters. Otherwise, you can't control the entire formulations without applications of statistics. So statistical applications is very, very important to get your optimized product. By heat and trials, if you say, this is my product, I got the optimized product, this one, nobody will accept it without proof statistically. So that's why, across the globe, different scientists given the concept of optimization, definitions of optimization in different way. So statistical optimizations is just by applications of experimental design in order to minimize your time, or you can cost efficient to develop your quality performance of your product according to Abu Isaac et al. Similarly, different scientists given there are different concepts according to their point of views, how optimizations is rationally or globally applicable by basically drug delivery system, or by analytical product development, or by synthetic chemistry, optimizations process during the synthesis of your lead compounds, or in green synthesis, or in combinatorial chemistries, or in pharmacological research. Everywhere the DoE concept you may apply best your vital or QbD elements. What are the target profile? What is your CPP? What is your critical process parameter? What is your CMA, critical material attributes? Based on your all the QbD parameters, you can select your different type of delivery systems. Hello. Yeah. So what are the merits of DoE techniques? I already told you. Few of them I will discuss, because it required fewer experiments to achieve the optimum formulations. No need to go for 100 trials, I already told you. And by DoE you can find out the best possible solutions among the 100 errors, and you can trace out and you can rectify your problems during the manufacturing or formulations and development process. And you can find out the important variables and unimportant, which is not relevant to your process developments. And lastly, you can minimize your errors by discarding the bad data, or which data is required, that data you can fix it, and remaining data no need to show, because the data is not having any relevancies towards your product performance. Similarly, so many things you can take your own decisions. You can find out, facilitate the decisions making before next experimentations by response mapping, by 2D or 3D response score, or so many other things are there. I will show you in my later slides. I told you that he is the first person, he is the quality expert persons, that is, Jordan, and he is the persons develop, quality by designs concept. Firstly, that is, quality could be planned in the first place to avoid quality crisis by building quality in the product. If you want to get your quality product, then definitely you have to apply QbD concept. So QbD is having the vital rules in every product development process. So he is the first person given the concept of QbD is globally in any sector, irrespective to get your product performance, desired product performance and robust formulations. QbD is mostly applicable in formulations developments. In FBD, what formulations you are developing in your lab, in your college, in your institutions, or in your small scale, process developments. If you are not sending your product in exhibit batch, or or not into the commercial final commercial product developments, then QbD, without applications of QbD, you can't send into your product into the market. But QbD is applications increase your scale of folds from 1 fold to 10 folds or 100 folds because of product performance, consumer acceptance, and there is no skepticism among the consumers. So generations of your product in lab or pilot scale is really squeeze because of without applications. And here, no regulatory applications, no regulatory issues in case of laboratories. But if you go for industries, in industry point of view, QbD optimizations concept is required for confirmations about your quality products to send your product commercially available in different market. So verifications is required if your product you are developing in your lab, and again you are going for technology transfer by getting your product for grant. After getting your grant for product developments, tie up with your industries, certain verifications is required. Really, whatever the product you develop in your lab, it is applicable by QbD or not. If QbD is not applicable in your previous formulations optimizations techniques, then nobody will accept it. So that's why QbD is having new or vital schools, because culturings encompassing quality principles and strong compliance to it. Because there is no regulatory issues and regulatory problems. So QbD is giving best informations to the customers that what branded products available in the market, that product branded due to the branded companies, and the companies definitely adopted certain tools. By using the tools, they have developed good quality products. So whatever the product is available in the market, that is QbD based product only. And you don't think that without QbD applications, anybody can get the product and send it to the market, and anybody can get without clinicals or preclinical trials. So what are the objectives of QbDs? The first objective is that quality excellence by QbD applications. You can satisfy your customer or consumers. You can produce number of batches within short span of time. Foods of tablet, foods of drug delivery system, you can develop by quality by designs concept. Best quality, best price you'll get. So in order to applications of QbDs, you can minimize your cost, minimize your efforts, and low risks. And you can, time is very, very crucial nowadays. Time, money, and efforts. Any industry, any sector, if you are doing your work honestly, if you are doing your work within the stipulated time, your target time, and you are sharing your knowledge to your team or your surroundings, definitely people will respect you, people will give judgment towards you, and you'll develop your product within short span, the quality products. Without you can get your profit margins in a better way, and you never face any kind of problems during your formulations optimizations. But dear friends, your delegates, applications of optimizations of QbDs always depends on 5 steps. So what are the steps of QbD approach or optimizations? Basically, in FBD point of view, formulations by designs, the first step is that why you are developing such formulations, whether it is a tablet technology product, or it is a semi-solid or liquid orals or self or gastro or micro needles or nanosponge or nanoparticulate drug delivery system or macroparticulates, whatever it may be. The first thing is that what is your objective to develop such delivery system, and why you want to select such drugs, or what are the selections criteria for such delivery system, about the excipients. Objective is very, very important. Next, after selecting your drug or excipient, when we go for combinations, after pre-formulation studies, heat and trial formulations, then you have to select your influential input as well as response variables. What are the independent and dependent variables? Basically, playing a crucial role during the formulations development, initial trial batches formulations. Next, you can develop your formulations of preliminary trials. Then you apply the design and you optimize the design parameters, and by 2D or 3D or counter plots or response curve, or by overlay plots or bounce hunter plots, or whatever the plots are available in the design software, you have to analyze it and you find it the significance, observations, and optimize your formulations. But before saying these optimizations, you have to prove it under what basis you are saying this product or this batch, these formulations is optimized. If you are giving the concept by analysis, by statistical way, by F test or by student test or statistical parameters with significance or not significance way, then only you can say your product matching your getting your objective, preliminary objective, setting objective. Then you go for scale off or commercial batch productions, or you can go for patent if you are academics or industries, or you can submit your product to the FDA for regulatory approvals. And after that, immediately you produce your product with crores of your drug delivery system with having desired quality performance in a production way. So FBD by experimental designs, that informations required obtaining the efficiently or precisely as possible without changing your objectives, just for applications of statistical design, by making your size of your formulations with limited number of trials, limited number of experiments, and applications of QbD suitable software, which type of software is fitting to what type of delivery system. That also very, very important. I'll show you in my later slide what kind of delivery system, how you'll choose the experimental design software based on variables. So these are the experimental designs. Basically, it is two types. Initially, you have to go for screening designs. Later on, you can systematically optimize by the applications of response surface designs. So if you go for formulations by design, you can go for screening designs and followed by response surface design. Screening designs, maybe you can apply for optimal designs, fractional factorial design, or you may apply for Tagosi designs, Blackett-Warman designs, so many other screening designs also available. You can apply any suitable screening designs, and based on your influential variables to screen your influential variables. That influential variables, again you fix the coded values of low levels, medium level, and high level. Then you go for systematic optimizations. That is by the applications of response surface designs, whether by central composite designs, because most of the scientists, globally, they are applying such design, that is composite design, or central composite design, or fractional factorial design, factorial design, or box Bentham design, or simplex measure D optimal design, I optimal design. So these are commonly used as response surface design. So many other designs also available, because it is not possible to write all the designs which is suitably applicable or rationally applicable for most of the delivery system during the formulations optimizations. But your friends, once you are applying the drug delivery system, basically in the applications of optimization process, you should know the concept of design space. But what actually the design space? When you will plot in your software, you will get 2D, 3D plots. Then how can you identify all this plot? What actually the control space? What is design space? What is the explorable space, and how it is important during the product development process by the applications of DoE. Dear friends, to get your assured quality products, you have to screen the different process parameters or different material attributes, or you can screen out the different QbD parameters. So that's why design space concept is highly applicable in case of drug delivery system. If you apply the design space, then you can movement. So for example, you fix any concentrations of any polymer from 10 mg to 100 mg. So this is your range, 10 mg to 100 mg. You play with your variations of your different formulations by changing 10 to 100. Don't go for below 10 or above 100, because you are fixing from the preliminary trials, you got some certain observations. And when you are fixing the low level and high level, within the design space, then you can find out the control space where exactly you are getting your desired responses according to your target profile, QTDP, that is quality target product profiles. So the movement always between 10 to 100. What you are fixing, that is within the design space. Movement is only possible, but once you are beyond this, That is, that is not your within your knowledge. That is completely explorable. That is blank. You don't know what will happen if you change the polymer concentration. Maybe you will not get the sustained product if you are preparing a sustained drug delivery system or control release product. Suppose instead of HPMC, if you are selecting different polymer PVP or PEZ, maybe instead of retarding the drug release, maybe more faster the drug release. So always you have to fix the concentrations, the coded values of your independent variables within the design space or within your knowledge, not beyond your knowledge. Dear friends, dear delegates, control space is very, very important. And within the control space, you can play anything. But if you are changing, then there is a loss of excipients, loss of solvents, loss of resources, time, efforts, and the company will, within 1 second, click you and remove you any organization because you're wasting our time, wasting our product, wasting our monies. And because why company is very, very cautious during this control space or design space, in order to get limited or minimize the regulatory, observations or comments given by the regulatory authority, whether FDA or ICH or NDA filing or ANDA filings. So that's why always be happy with your control space or design space. If anything, after your design space or control space or just periphery of your knowledge space, you discard such product, you remove such formulations, no need to go for again any trials for the trials, because you already set your critical process parameter. My critical process parameter is this, HPMC concentrations. My critical material attributes is this. My CPP is a percentage of drug release, percentage of drug content, percentage of my microadhesions, zeta potential values, or self-emulsifications time. This is my CPP, for example, and my concentrations of X polymer, Y polymer, Z polymers, and what is the impact on this. After that, if you are getting according to your CPP values or according to your desired goal, then you can fix it because you are getting within your target or within your true optimum, within your design space, within your control space. But anything you are beyond this, beyond your selections, that you can say this is the edge failures, because you are evaluating when you are formulating something, you are going for characterizations. You are checking the drug release studies, you are checking for drug content, swelling index, polydispersibility index, self-emulsification time, whatever the characteristics parameter according to the delivery system, all you are evaluating. But what exactly you are getting? No, you are not getting your as per your target. Then that indicate it is the edge failures. So the edge failure is always considered based on your evaluations, based on your specifications, based on your observations data. Okay, so edge failures never consider your formulations. So the importance of design space to minimize the quality risks, to understand about the interactions among the critical material attributes, critical formulations attributes, and you can validate your product by the applications of design space, and you can get your full scale operations about your product optimized product, because you can find out your design space. If I'll see this is my area, I want to see it. If I'll see it somewhere else, there's a chance of infections. So that's why I'm sitting in one place. This is my true optimum or design space area. Within this level, I can change my concentrations of polymers. Then only I'll get the true optimum, final optimum, optimized products. Within the design space, you can fix low level and high levels. Medium automatically you will get. So dear friends, you can find out how the design space is applicable in quality by designs. Without finding the control space, if it is beyond your knowledge, or then you can discard it. You have to get ideally movements for the product developments for full scales. So these are the few examples. This red area is indicates the point of failure because it's showing the edge failure of the formulations. And this is your explorable space. The entire, like circle, it's egg-like forms. This is the explorable space. Within the space, you can change your level of concentrations of your polymers, selected polymers during your drug delivery system. And this square This green, area shows the point of success. This is your optimum. This is your true optimum. You can say, this is my success product because I got percentage of swelling is highest in case of these formulations. You are writing in the concluding remarks of your manuscript, of your articles, in your thesis, or you are, project report. This is my final or true optimum because of this characteristics or this CPP values. And this because of my target product profiles and it not having any compliance related to regulatory issues and giving best results, optimum operating results with a simplicity way, in good representations way, and I got a benefits among the other delivery systems or other formulations batches. So that's why I'm predicting this is my design space area, design space, level by which I can fix my formulations batch. And if you see, the expansion of a possibility or not, sometimes what we'll do, you can ask to your boss or your supervisors, PhD supervisors, or your researchers, or the team or lead, sir, can we change little bit? There is a question mark. Why you want to change? If you want to change the concentrations from, 100 to 105, what is the variations you are getting? If not significantly changed, then you can change. Probability also there, but it is case to case, delivery to delivery system. Sometimes efficacy you want 100%, but you are getting 19%. That time you can change it. That is case variations. Certain case, if you apply your original design space artificially, you can constrain for simplicity way. But sometimes if you want to expand beyond your knowledge space, what is the changes in the CPP values, critical process parametric values, or your responses? If gradually changing, then you don't change or expand your design space levels, because you may not get, change in temperature. For example, change in temperature, one parameter, independent parameters. What is the percentage of seed weight? Yield values. For example, you are preparing capsules, micro capsules. You are changing the concentrations of some udrazate polymers. What is the yield value? If the yield value is gradually decreasing by increment of changing of your concentration of polymer, you don't go for expanding your design space or your true optimum levels. Always you fix it depending upon your concentrations and the characteristics. And there is a chance of edge failure more if you're expanding your changing of your design space concept. So this is about the different cases. No need to discuss this. And always you fix the design space, and within the control space, I already told you there is no regulatory problems, no regulatory submission. If you are preparing in your lab, may not required every time to control your design space, but if you apply, I think it is a very good scientist or very good researchers, they can say, okay, you are within the true optimum levels. You may go for patent filing, or you may get, there is a chance of, according to your claims, according to your innovations, your backgrounds, you can protect your claims as an innovations, right? So regulatory submissions not at all required, sometimes. But if you go for true, if you are going for beyond your true optimum or design space, regulatory submissions, regulatory issues, definitely you'll get during your product developments. So that's why always fix your design space knowledge characteristics. In regulatory importance point of view, design space are three types. One is lab scale design space, controls, that is pilot scale design space, commercial scale design space. Lab scale means what you are developing. No regulatory importance. Regulatory persons, they will not bother about your product development, what you are formulating, because sometimes people are manipulating. They're saying, sir, I have added this, or we have added this, and we got this good product. But reality, nobody is seeing. Nobody is seeing the original data. This is completely factual data, not factual data. If you get the factual data, then all you'll say it is really within the design space. But actually, there is no regulatory importance if you are developing in your lab. But when you go for commercial design space, not in the intermediate also. In intermediate also not required, no regulatory importance. in between, small scale or large scales. In technology case, technology transfer case, in pilot scale designs, not required design space. So the design space is always useful, meaning regulatory importance in case of commercial product developments. So prior knowledge is required. What are the developmental studies you are applying? And you can understand the product, what you are developing by design space, because you will get in depth about your formulations. You can understand your process. If anybody will ask any questions, how you formulate, what is your processing steps, immediately you can say. But those persons, they are not applying such things. They will, they have no voice to open how to start the works, how to select the excipient, how to select the drugs, and what are the steps they have followed, how they have optimized the processing parameters, what are the quality target product profiles, and how they have analytically applied by the risks associated with the QbDs with the applications of PAID, that is process analytical tools for minimizing the risks. So the entire things can enhance your understanding about the product and the process by the concept of design space, and you can improve continuously about your skills related to formulations, and you can change anything about your regulatory problems and within the design space, and whatever the verifications is required during the manufacturing operations, you will change it so that your manufacturing experience can enhance. So pharmaceutical developments from exit batch to commercializations, you can keep in your mind up to the end. Don't think that I'll keep always at the end why it is failures. No, from the very beginning, you are very pertinent during your initial screening process or screening designs process or screening designs of your applications in your mind that no design space I need. I want true optimums in order to get to my quality products, in order to optimize product in a robust way with better efficacies. If I'll say one example, one case studies, there is one CQA, that is one critical quality attributes, for example, dissolutions, percentage of dissolutions. Two CFA, that is two critical formulations attributes for parameter one, the concentration of let us HPMC and concentration of EC, that is parameter two, and then how it is changing the percentage. This is the 2D, 3D plot. This is the 2D plots. This is the counter plots. This is the response plots. And how it is, dear friends, you see the color, the different colors is indicates the explorable space, design space, control space, true optimum space, knowledgeable space. So always you think this is our design space, and within the design space we're getting the true optimum space or true space, and where we can find the best quality product or quality developed products. And whatever these things, there is no significant influence on the CQA, that is critical quality attributes. Similarly, if you compare, two critical quality attributes like percentage of friability in case of tablet dosage forms and dissolutions, a percentage of drug release, and how the combinations of these two critical quality attributes with the two parameters affect with the two parameter one and parameter two, the combined design space you will get like this. And this combined design space you'll get in the overlay plot. This is one kind of overlay plot between the 2D and 3D to find out the exact true optimum design space where both the influential variable of parameter 1 and parameter 2 impact on the responses, two responses in case of tablet dosage form, that is percentage of drug release or the dissolutions characteristics as well as the friabilities. So if you go for other cases, you'll go for 4 CQAs, 4 responses. For example, release, percentage of drug release after 20% of drug release, or zeta potential in case of self-emulsifying drug delivery system, or you can find out the percentage, entrapment efficiency of the drug, or if you want to, what is your target product profile related to the particle size, because you are preparing a micro or nano, whether within the nano range or not, within the 100 nanometer or not. If not, then you can't, fix your parameter of whatever the excipient you have selected, like maybe competitors or queen or whatever the 2 CFA you have changed that according to the 2D and the 3D plots, you can find out, the design space or true optimum concentration of the selected CFA. You see how you are getting the CFA. This is your true optimum. Within this, you can change your concentrations of competitors and P90s in order to get the desired characteristics of your zeta potential values as well as release of the drugs. So this is the combined, 4 CQAs and 2 CFAs. And this showing the size, this is automatically you'll get in the software based on your fixing responses as well as the label coded labels, and will get within your target product profiles. So these are the 4-point strategies. So this is the implications of QbDs for product developments, improve the efficacy of method to test bioequivalence, and develop method to assess bioequivalence locally acting drugs, develop method to characterize complex drug substances. Dear friends, this is nothing but FDA 4-point strategies. When you are applying your quality product developed by your industry or in your lab, then you go for submission into the FDA for regulatory issues, any regulatory issues. The FDA, the first question is whether you have applied in your product development process of 4 strategy or not. The implications of QbD is the first. You see, implications of QbD, if you are applying in your product development, then you are applying the first strategy. The second one is because of implications of QbD, you are improving the efficacy of the drug product or not. And whatever the test product you are developed, have you compared with the marketed product or not? And how it is bioequivalent with your product or not? Have you performed the bioanalytical studies or not? That is also important. This is nothing but FDA need this 4 strategy. And the developed method, analytical method, bioanalytical method, what you have assessed in your bioequivalent studies for locally acting drugs or systematically acting drugs, then you have to check it. Really you got the improvement of bioavailability or not. If there is an improvement of your test dosage form with the existing marketed product or available product in the market commercial base, then definitely you can get your product in a robust methods. And the method optimizations, characterizing your complex drug substances with the applications of QbD and the development of analytical method by serum or by blood or by HPLC or EPLC or any sophisticated spectroscopic or chromatographic techniques, then definitely that during the bioanalytical techniques also you have to apply the QbD concept, like concentrations of your mobile phase based on solvent ratios, compositions of your mobile phase, run time, retention time, theoretical plates, etc. You have to fix it. Once you are optimizing the method development, then you go for bioanalytical studies for bioequivalence analysis of about your test products, and then later on you can go for IV correlationship, or before that you can perform the permeability studies in intestinal perfusion studies about your drugs. So this is nothing but FDA 4-point strategies. Otherwise, there is a lot of issues related to your quality products. So these are some of the computer are used in systematic QbD or formulations by designs in different drug delivery systems. In order to get a suitable, robust, optimized drug delivery system, computer is essential. You don't think that by verbally you have to apply, or you have to say something this. I'll fix it and I'll write it. Computer is very important. Software installations is required. Purchase software, is required in order to get a quality results. And don't go for trial versions. Maybe for understanding, you can start with trials, not by systematic optimizations with directly applications of the licensed versions. You have to appear in a number of presentations related to this type of webinar or seminar or faculty development programs, etc. To get your optimized drug delivery system by computer assisted data analysis without applications of DoE, design of experiment, during your drug delivery systems or during your experimental design process, you may not get any success, maybe failure. You may publish volume 1, issue 1 journal, but you never published any standard or impact journals. In order to get the JCR indexing, SCI expanded journals, or the high impact, high indexing, Scopus indexing journals, at least you have to do certain bioanalytical studies or QbD applications in your formulations developments to get good results, good products, good innovative thesis, innovative presentations, and innovative, work you have planned in your futures. So the design experiments, these are the few of the software available freely as well as paid versions. Design export, Umetrics, Minitab, JMP, Statisticals, and Stat Graphics. So many other types are there, like Multisimplex, Factors, SAS, SPSS, Systops, etc. So many things are there. Yeah, what are the sources for getting the desired, type of your delivery systems of different types of software, and there are web sources I'm showing in these presentations. If you want, you can take the screenshot, there is no problem. And the JMP, Fusion Pros, and these are, free software available trial versions for 15 days, 1 month, 2 months. Then later on, you have to register it, and the paid versions, you have to subscribe for your day-to-day activities or your formulations optimization process. So many other paid versions are available, like, DoE, PCI, IV, Statistica, Nemrods, Modis, DO, Wisdoms. Others are there: Extarts, Optimans, Compact, Omega, ICIDs, Solvers, Matrix, and any drug delivery systems, whatever the drug delivery system, one variable at a times you don't approach. Don't select only single variables. You screen out the multiple variables in your formulations development process. Then only you can find out what type of independent variable you want to select. Because when you are preparing any kind of dosage form, for example, you are preparing tablet dosage form, don't think only one concentrations having several impact on different CPA, that is critical formulations, critical process parameters. You have to find out each and every excipient's influence. What is the concentrations? What is the impact of diluent on this? What is the impact of lubricant on this? What is the impact of super disintegrant on this? What is the impact of direct controlling polymer on drug release? Everything you have to screen out. Don't fix one variable at a time. You may optimize in later stage after applying preliminary screening designs. Nowadays, whatever the delivery systems are available in the literatures, based on the literature's level, applications, I find out that most of the scientists, basically, sustained release product, they have used optimizations techniques, sustained release product, most of the time they're applying. Then later macro particulate drug delivery systems, then microspheres or other delivery systems. And if you'll find out which type of design experiment scientists they have applied according to the available literatures of till a day before astro days, the factorial designs, most of the scientists they have applied. Why? Because it is easy, and the advantages is much more simpler than other designs. And later on, people are applying box Bingham designs. BBD, that is a box Bentham design, then central composite design, then de-optimal designs, then like that a simplex design, Latin square designs, simplex mixture design, like the Tago's design, or etc. Other designs that are applicable based on the influential variables. And if we'll compare up to 2020, oral drug delivery systems by non-oral drug delivery systems, as for the last 6 decades, novel drug delivery system, the literature reports showing that, oral drug delivery systems, applicability of DoE or QbDs or FBDs optimization techniques is much more, every year, every decade, there is increasing the applicability of DoE concept. Is a very good science. And the number of response literatures versus, number of response variables, gradually there is a decreasing, because people are screening in better way. Scientists are screening, researchers, they are screening in better way in order to find the desired data profiles. And I told you these are the different delivery system, what type of delivery system, and what are the process variables, independent vectors you want to select. For example, if you will go for micro particulate delivery system, then process variable you may select nature of mixing of the vessels. You may go for selections of speed of steering or steering rate or steering time or durations of cross-linking process, injections rate. These are the probable, probable microparticulate drug delivery system, basically for independent variables. But what are the response variable you want to select for such delivery system, like, in vitro drug release, or you may go for entrapment efficiency, percentage of yield of the drugs, sorry, delivery system. So, and what are the feasibility or best choice of design of experiment, like factorial design, fractional factorial design, central composite design, box Bentham design, simplex mixture design, etc. Like that, vascular designs, what are the independent factors, what are the response variables, and how you want to select delivery system. Osmotic drug delivery system, like, independent variable for osmotic drug delivery system, polymer content is very, very important. Orifice size is very, very important. Coating level you have to select. So many other parameters there. Just I'm giving few examples based on the literatures. And these things always, keep in your minds. You set this particular, level by which you can get some kind of optimum concentration or optimum value about your optimum formulation. And if specific, most of the literatures they're applying in case of osmotic drug delivery system, that is fractional designs, sorry, factorial designs, as well as box Bentham designs. Like that, we have certain objectives in case of, self-emulsifying drug delivery system. Basically, we are selecting low bioavailable drugs, low permeable drugs, and with also low soluble drugs in order to achieve, increasing the solubility, improvement of drug solubility and drug viabilities, in systemic circulations, or improving the permeability through the tissues, permeability to the perfusion site or in the target sites, or tissue perfusions, or other site of the our compartments. So then we'll go for different type of delivery system, different types of design based on the responses. We have to fix it. And the quality, quantity, so not quality, quantity of ingredients, solvent also sometimes you can consider in case of self-emulsifying drug delivery system. It's very, very important. Nobody is selecting, rarely few literatures available so far. Content of oil, concentrations of, solvent, sorry, volume of the solvent, nobody is selecting. Sometimes the volume may change the characteristics of your desired responses, like transdermals, semi-solid drug delivery systems, vesicular drug delivery system, microspheres, like homogenization speed, mixing time, mixture speed, response you can select, right? Entrapment efficiency, drug contained, swelling index, moisture index, etc. So many things are there during your characterizations part. What are the characterizations parameter you are selecting? This is one response you have to select. Later on, you can fix what are the real, influence changing based on the changing the coded value of your independent variables. If you are getting this type of, locations or, stationary point in your 3D plots, then you can say this type of plot is showing the response having maximum response. If you are getting the 3D curve like this is the minimum response according to change in the parameter of independent and dependence. Sorry, I'm beyond the time actually, 10 minutes late because of, some important things not covered up. Sorry for taking the time. I apologize for that. So if like this, you are getting, this is the settle points, this is the stationary reach of response, and this is the rising reach of your response gradually by changing the variables of X1 and X2. And if I will say one case, or, percentage of drug release with respect to time, how, if your target is within, 9.11 hour, I want the percentage of drug release to be 60%. Then according to your response values, the feasibility search of optimized formulations with your labels, this is the, table value data. The yellow color indicates our, this is our QTTP. This is that our target product profiles. We want within 9.11 hours, 60% of drug release. And based on the graph, we have fixed this. But in the formulations, initial trial batch, initial, not trial batch, in the response surface of optimized formulations, where exactly we are getting. If you see the parameter of low level -1 and the high level, we are getting 9.11 values. In other cases, you see there is no 9.11 values. Then you can fix, this is our true optimum formulation. Selections of formulations based on your table data, optimized data. Then you fix it -1 concentrations of parameter 1, concentration of polymer 1, concentration of highest concentration of polymer 2, will get the 60% of drug release within 9.11. 9.11. If I go for one of the case studies, that is self-emulsifying drug delivery system, then you see the preliminary requirement of any self-emulsifying drug delivery system for industrial point of view. You always select the drug, lipid, then you can go for lipidic solutions, and you add with certain specific emulsifiers, and there is the optional for you during self-emulsifying, whether it is a micro or nano co-emulsifier you may add or you may not add. So that is getting the self-emulsifying drug delivery system. And if you go for dilutions, then you can get for nano or micro according to the range, right? And after that, you can go for ternary phase diagram studies by applying the software, ternary phase diagram software, Prism software or something, and you can evaluate based on the emulsification range or areas where you are getting the maximum micro emulsions or micro gels or micro emulsions area according to the color, according to the thing, selections of your label in the software. Suppose you are fixing in the initially this color is for micro emulsions, then according to the number of formation of microglobules or nanoparticulate system or microparticulate system, such concentration, such ratio, you can fix for further preparations of self or nano emulsification drug delivery systems for optimizations process by the applications of QbD or FbD or other CCD or whatever the designs. This concentrations ternary phase diagrams is very, very important in case of self-emulsifying drug delivery system, to fix the ratio, whether this ratio is good or this ratio is good, whether 1:1 ratio co-mixture is good or 3:1 ratio or any other ratios. You have to titrations method, water titrations method, ternary phase diagrams is initially important to predict the which type of drug delivery system, which type of self-emulsions, formulations, mixture showing better results in order to go for further preparations of nano or micro. Then you can go for characterization studies like mean percentage of drug release with respect to time and of different formulations batch, your optimized batch with your pure drug, what the drug you have selected. For example, carvedilol you have selected, your pure drugs is this, and you have prepared, liquid self-emulsifying drug delivery system and solid self-emulsifying drug delivery systems. And you compare it, in which case the mean percentage of drug release is better. According to that, you can find out these formulations is showing better results as compared to marketed existing products or as compared to pure drug, as the pure drug is very, very, showing a little solubility is characteristics, insoluble in water, but after converting into a self or nano or liquid self nano emulsifying drug delivery system, showing better results. And similarly, dissolution efficiency, you can predict in your optimizations 2D or 3D graphs. Liquefaction time is one of the response. Emulsification time is another response, where the exact response you are getting MDP, mean dissolution times, and permeations rate by interstitial permeations rate. Then you can overlay plot, combine plots, where exactly you are getting the better emulsification time, liquefaction time, MDT values, percentage of drug release with the variations with the coded value of X and Y with the concentrations of polymer 1 and polymer 2 like liquid 1 to 27 for self-emulsifying drug delivery systems. But you have to do certain characteristic study like X interstitial perfusion studies, or you may go how much the drug perfused to the intestine of the suitable models, what model you have selected after bioanalytical validations process. Then only you can find out the intrinsic permeability, effective permeabilities, and warp permeabilities. According to your product, how your product is better in case of wall permeabilities as compared to intrinsics versus effective permeabilities with respect to available marketed products, whether it is exclusion conflicts or it may be a marketed products. Then similarly, absorption parameter also you can find out, and absorption number, fraction dose observed, everything you have to determine during the characterizations analysis to go for in vitro and in vivo studies. And stability is very, very important for industrial point of view or industrial milieu or industrial perspectives. And in vivo pharmacokinetic studies by your suitable animal ethical approvals data, and which formulations, what you are saying that this formulation is my optimized batch, then you have to show in pharmacokinetic analysis by your suitable analytical models my liquid self-emulsifying drug delivery system showing better viabilities in the blood or in the serum as compared to pure drug or marketed products. So similarly, you have to compare with all the pharmacokinetics parameters with respect to the different drug release product available in the market. Also, you can compare with your pure drugs. Then influential value of rate constant, Cmax, AUC, last, everything you can find out. If this value you are getting, then you can say my product is showing, my optimized base is showing better AUC, better rate constants and Cmax values achieved within limited times as compared to marketed or pure drugs. Then you go for in vitro and in vivo correlationships with the different level A, level B, level C, or level D, or multiple correlations with the in vitro drug release, drug percentage of drug release versus in vivo viabilities. You have to compare it. If you are getting the correlations, R-square values is 1, then you can select such correlations level compared to others. And these are the self-emulsifying drug delivery systems available in the market, manufactured by different pharmaceutical companies with different trade name or brand names based on their solubility characteristics they have selected, and later on after QbDs or optimizations doing certain characteristic studies, and they got the solubility improvement, viability improvements, and this. And lastly, I want to show a few of my research publications related to this, QbD based formulations based of my and my team research teams published so far. This is my brief profiles about the Google Scholars, right? And these are the publications in the form of, book chapter in Springers or in elsewhere. And these are my publications in, Science Directs and chapters. And this is my recent books, computer-aided drug developments, right? This is my patent. Okay, this is my patent grant, what I have already placed for technology transfer. And this is my published patents I already filed for grant. And this is my research publications recently 2021. And these are my previous publications with my research team and my pursuing PhD students. Okay, so these are other publications of review articles or in the research articles, right? With high impact journals. This is my previous publications. In Springers, Bentham, Informers, other standard Scopus or UGC listed journals. And this is one of the recent articles I published, Quality by Design's Prospective in Pharmaceutical Applications in Double Emulsions Techniques. So I am the corresponding author for these publications. This is the nanoparticulate iprosatans mycelates and estromoprazoles magnesiums, iribistatins drug delivery system, microemulsifying. This is only I am showing the microparticulate drug delivery system or self-emulsifying drug delivery systems. These are the other, and this is my award I received. And my concluding remarks about my presentations, QbD, I already told you, is always giving the first place for the pharmaceutical product developments. It's considered the best approach in order to get quality product for the benefit of the patients, not for us. So QbD is always applicable for improving the life cycles of the pharmaceutical products, and it is a versatile tools for applications of QbDs to get minimize your toxics or ADR monitoring of your drug product to improve the efficacies and safety profiles because of consumer demand, consumer needs. And an QbD approach enables the manufacturing standards and identifying to understand the variabilities during the process developments and establish the control delivery systems to get the quality of the product. So QbD's, get our goals, maintain our targets, and develop our pharmaceutical products with the minimizing the errors or associated risks based on the regulatory issues. These are my few references during the preparations of slides. And lastly, again, I want to add my proud privilege and expressed and heartfelt gratitude to the management of Nirmala College of Pharmacies, Dr. Sheikh Abdul Rahman sir, for giving me these opportunities, as well as I grateful to the management of Nirmala Group of Institutions for encouragements and selecting the different resource persons. And lastly, my sincere thanks to Atul AICTs for sponsoring such e-faculty developing programs to encourage our young and budding pharmacy professionals in the field of different delivery system or in different sectors. Really, it is a very good platform for me to share my knowledge. And at last, I am very much thankful to the coordinators, Dr. Reddy, Pomala Reddy, Art Pomala Reddy, for inviting and arranging such things. I wish all the best for all the entire Nirmala teams for your great futures. And I have seen such a dynamic principle to motivating to the faculties and management for developing the infrastructures in the form of quality education to the students and thinking about the students' future. Really, it is a grateful, sir. Thank you very much, each and everyone, for giving me the opportunity. Stay home, stay safe. And if you have any queries, you can raise your questions. I am extremely, apologizing for beyond your times, your valuable times. And if you have any questions, you can raise your questions. And if you want to connect me, this is my, details. Thank you very much. If you have any questions, you may raise the questions. Thank you.
Thank you, sir. Thank you very much for your wonderful presentation, sir. Now I request you to address the queries raised by our delegates. Dear delegates, if you are having any queries, you can post it in the chat box. Once again, I request, dear delegates, if you are having any questions, please post it in the chat box.
Thank you, Dr. Gauri Dixit, for your nice remarks about my presentation. Thank you very much.
Now I think we are on the verge of ending this session successfully. I, Sudhir Madhyala, Associate Professor, on behalf of Department of Pharmaceutics, Nirmala College of Pharmacy, extend my sincere thanks to Dr. Suryakant Swain sir for your valuable time and sharing your knowledge to our participants. Now, I would like to invite Dr. B. Pamula Reddy, Organizing Secretary, to honor the resource person.
Thank you, sir. Thank you, Sudhir, sir. So, on behalf of Nirmala College of Pharmacy and AICT Atal Academy, Dr. Suryakant Swain, sir, we are very thankful for your valuable information. I hope all the participants have received the wonderful words from this session. I hope all the participants will implement this session in their career growth. We request you to accept our token of gratitude in the form of e-certificate. Thank you, sir. Thank you for the wonderful session. Please accept our request.
Yeah, thank you, Pamela Reddy sir, for your nice gestures. But at last, before ending these sessions, without beautiful participants and delegates, I never think that my session completed with good remarks. And thank you very much, dear participants and delegates, for patiently you are listening my entire sessions. Thank you, everyone. Thank you very much.
Thank you, sir. Thank you very much, sir. So thank you all the participants. So we'll meet at 2 o'clock, session 9. Okay, thank you, one and all.
Thank you, sir.
Yes, sir.